Tac. All right, I think we're live. Hi, everyone. Welcome back to the Worldwide Neurodevelopmental Forum. Hope you had a good summer break. For those of you who had one, it's a great pleasure today to start this new academic year, I guess, with Claire Viart. Hi, Claire.
Hi everyone.
Welcome online. Live from Paris. I'm a bit rusty too, in fact. I'm clicking on my windows here. Yes, so Claire, a little bio, and then the stage will be fully yours. So Claire Viart obtained her PhD in biophysics and neuroscience. In terms of biophysics, she did kind of some hardcore stuff, working on neurons in vitro, trying to figure out how activity patterns neuron in terms of their connectivity. That was in Strasbourg. And then she went for a postdoc in Berkeley, where she started with her main interest, which is in vivo live imaging. And in particular, she was working there on approaches that involved optogenetics in zebrafish larvae. Came back to France and started her own lab at the Paris Brain Institute, where she since then has been focusing on understanding a sensory motor coordination in zebrafish, which in particular some very interesting and original work on the role of some centrally located neurons or cell subtypes that act, at least in part, and I think for many other purposes, as mechanoreceptors. I think we'll hear quite a bit about that. Claire is passionate about her work, like many of us, but she pushes the passion as far as imitating her model animal. The word comes from her. She's passionate about swimming, not only swimming, but swimming in cold water and against the stream. So I think we'll stop there. Claire, great to have you for this first new session, and looking forward to your talk.
Thank you, Denis. Thank you very much. It's really an honor to be here. And it's a real pleasure to get to connect. So I'm going to share. You guys see my screen?
All good.
Great. Cool. So I will present the work that we've done today all over almost the last 10 years now. And I will try to really give credit to the great people who I've been lucky to work with, who have been really fantastic to figure out this new sensory pathway in the vertebrate spinal cord. So, changing slide problem. Okay, so the classical view on sensory feedback is that the sensory system is organized in the periphery, and dorsal root ganglia bring sensory information to the spinal cord that gets integrated directly and indirectly by motor neurons. And this sensory motor loop are critical for adjusting locomotion and includes proprioception. So we and others got really interested in the last decade, and I know that Guillermo Lanuza and Nicolas van der Berg are here, so I'm really excited to have a discussion with them. We got excited by rediscovering cells that look like sensory cells. They're very similar to hair cells in the first approach that were described by Colmer and Agdur a century ago in this kind of drawings when they were labeling sparsely these neurons, and they noticed that they had an apical extension bathing in the cerebrospinal fluid, and also an axon that you can see on the left here. The two of them agreed that these cells look very much like sensory cells, but they had no way to really investigate their function. And they disagreed very strongly about how these cells could possibly interact with the Reissner Fiber, this polymer that you see with a black dot in the center here in the central canal, that had been described by Ernst Reissner already in 1860. So Ernst Reissner is looking a little upset here. He had been finding in 1860 by looking under the microscope at the spinal cord of lamprey that inside the central canal there was a very large acellular thread that he could see easily, and he drew as a straight line like this. And since then, it's been shown in many animal species that, in fact, the Reissner fiber is found in has been found in most vertebrate species, and the diameter is different. In all species, it's acellular. It's running freely in the central canal, and it looks like it could be interacting with possibly cilia. So we decided to revisit these questions, and Lydia Genoun, who I think is in the audience, has been the first student to really tackle this question and answer the first point, which is like, are these cells indeed conserved across vertebrates, and do they keep similar markers? So she found that these CSF contacting neurons were present in macaque by looking at 2 adult spinal cords at the cervical, thoracic, and lumbar level, and she found that they had a little bit of a cauliflower structure on the top, GABAergic neurons expressing different enzyme and transporter for GABA, and also a channel PKD2L1 that had been identified by Charles Zucker's team to be involved in sensing pH and osmolarity. And when we started, Nicolas van der Beek had already shown that in the mouse, these cells were indeed responding to pH and osmolarity and had a very strong activity of the PKD2L1 channel. So we looked in the mouse and we found indeed these cells starting at E14.5, and we find them also in zebrafish starting at early stage E18, 18 somites in the zebrafish embryo. So early development. And similarly, there are again GABAergic neurons with the PKD2L1 channel clearly located in the apical extension. So the Reissner fiber, we identified it, and this is the work of Yasmine Canto-Bellarif. We identified the Reissner fiber by using an antibody from Stéphane Gobron that recognized the material from the cow, and it works really well because it's a very conserved structure made of SCOspondin. It works really well to identify the fiber as well here in the zebrafish lab as seen from the side. So now we tackle the question of understanding the function of these cells, and in particular the mechanosensory function, by investigating mainly in zebrafish. And the reason is that the animal is transparent at the embryonic and larval stage. And so we generated different sets of transgenic lines using the PKD2L1 promoter in order to drive expression of actuators or sensors of activity in order to find out when are these cells active and what happens if we activate or silence them. And so this is taken from the work of Kevin Fiedlin. And we, using this line, could show that we have a very nice way to label these cells that form a little string in the spinal cord around the central canal. And if you zoom closely, you can see that they are indeed distributed on the 2 sides, left and right. And if you do mosaic labeling, which is, again, the work of Lydia Genoun, we found that their axon is running only ascending and ipsilateral. So we have an inhibitory cell with an apical extension projecting only toward the head, typically from 2 to 6 segments. They are very close to motor neurons, as you can see in this transaction here. So the CSF-CNs dorsal one are labeled by somatostatin, and the motor neurons are in green here. So what could these cells sense in the CSF, in the spinal cord, and how possibly can they modulate the activity of motor neurons nearby? So when we took a closer look at the apical extension with Lydia, we find that the apical extension is composed of one kinocilium labeled in green here, and numerous microvilli that are actin-based that surround this kinocilium. When we used APEX2, we confirmed that the cilium had a 9+2 organization. You can see the insertion of the beta body here. And imaging the apical extension both in vivo on this movie or in vitro in primary culture, we observed that indeed the kinocilium was actively moving. So what could be the function of this apical extension? And could it be, like Colmer and Agdur proposed, a sensory organ that could be sensing mechanical information in the spinal cord? Colmer suggested a third ear in the spinal cord. So we don't obviously, listen to music through our spinal cord, but could we be sensing spinal bending and curvature as a reflection of our posture? That's a question that Urs Bohm has been tackling, joining the lab after Lydia. And what we did with Urs is to induce passively and actively curvature of the tail. And we monitored at the same time the position of the CSF contacting neurons with TAG-RFP and their activity by using the sensor GCamp. So in this experiment, we have a tail bend induced by an acoustic stimulation, and then we can see an active tail bend. And we have other experiments where we come with a probe and push with a mechanical probe on a paralyzed animal. In both experiments, we find that with active or passive bending of the tail, we induce a recruitment of the CSF contacting neuron that is completely asymmetrical. So we observe that if we color code the left in red and yellow right, if we have a left tail bend, so the red cells are on the side of the bend, we see a massive activity in the cell on the side of the tail bend, so the concave side. And this is for the wild type. And in the mutant PKD2L1 that was generated by Lydia Genoun, we have an abolition of the response. So we don't see any more recruitment of these neurons. So Jenna Sternberg followed up on this work and used electrophysiology to record from these cells in vivo. And what she found is very similar to the work of Nicolas van der Berg in vitro in mouse, is that when you record from these cells in vivo, you have very high input resistance, so that in whole cell only, you're actually able to see single channel opening, as you can see on this image here in the wild type animals. Now, in the mutant knockout for PKD2L1 that we generated, there is no more activity of the channel. It's completely quiet. And what is remarkable is that a single channel is has such a large conductance. You can see here the scale is 10 picoamp in the center, that a single channel is sufficient to induce spiking in the cell. So here you see we inject 15 picoamp and we can induce action potentials. And the current is typically in the resting state around like 20 picoamp. So we've tried with Patrick Delmas in Marseille to do experiments when we put those cells in culture and come with a probe to push against the membrane and record the activity of the channel. And what we found is that very similarly to in vivo, at baseline, we have a spontaneous activity of the channel that you can see here. And when we come with a probe and push 4 micron against the membrane, so quite a good push, we observe this opening of the channel. So we increase the open probability of the channel, which leads to multiple channel opening. And this is not the case in the knockout for PKD2L1, where we don't have any spontaneous activity and we have no response to the push with a piezo probe against the membrane. So everything is pointing towards PKD2L1 is necessary for mechanoception in these cells, but what is the information that they're encoding in vivo? We initially thought that it could be CSF flow, and I'm going to show you the pieces of evidence that we have that are quite challenging this idea now. So we used 3 methods for measuring flow of cerebrospinal fluid in vivo. The first one on the top here is imaged with a spinning disk microscope, where we're looking at fluorescent particles that are little beads of 20 nanometer diameter. And as you can see, when we inject these beads in the hindbrain ventricles, they are flowing down in the central canal, and we can see the movement of these particles, where it's obvious that we have a lot of Brownian motion. But if you look carefully, you can maybe notice that the ventral particles tend to go from left to right, which is from rostral to caudal. the typical direction that we all assume is the CSF is flowing in the textbook. However, in the dorsal side, you can see that the particles are going the opposite direction. They're going from caudal to rostral. So we were quite puzzled by that. And we remarkably observed it for endogenous particles when we used a technique called dynamic full-field optical coherence tomography in collaboration with the lab of Claude Boccara and Olivier Touvenin, who was doing his PhD at that time. We imaged the embryonic CSF and find that in the CSF, without any labeling, we already have, a lot of particles, can play the movie again, that are flowing in the exact same dynamic that what we see for the beats. Sorry, I'm going to try to replay this movie again. Okay, so very similar dynamic. And recently, we used a third method for now making transgenic animals where we can label endogenous particles without any injection. We use CD63 fluorine, and we find drivers where we have particles labeled in the CSF. And as you can see on this movie from Yasmin Canto-Bellarif, we have a beautiful labeling of endogenous particles, possibly exosomes, that are showing the same pattern of strong Brownian motion and bidirectional flow. So to quantify this flow, we've done chymographs where instead of having a movie where we look at dorsal, ventral, rostrocaudal over time, we rotate our movie so that we can look at either dorsally or ventrally what is the typical trajectory of particles if time is on the y-axis here from 0 to 20 seconds and rostrocaudal is on the x-axis. And so what you can notice is that the particles are going from rostral to caudal on the ventral side over time, while on the dorsal side, the particles are going from caudal to rostral. So we really have bidirectional flow. So Olivier Touvenin implemented an automated method for quantifying this flow in order to be able to compare it across animals, across mutants in many different conditions. So it's an automated algorithm to just look all the way from the dorsalmost to the ventralmost side, and we can see that we have indeed this beautiful bidirectional flow. He studied then the origin of this flow and found out that the flow was due to beating of cilia. And so this is a movie from Andy Prendergast, now another postdoc in the lab, who quantified the beating of the cilia and find that only in the ventral side here are beating cilia. There are very few beating cilia on the dorsal side on the right. And if we image them now from a lateral view, as you can see on the right movie here, what you can see is that the cilia have a polarity that is directed toward the tail, and they are beating, again, mainly on the ventral side. So together with François Galler from the Physics and Engineering Department of EPFL, and in particular Ludovic Kaiser, a postdoc in his lab, we've been building a model to explain the bidirectionality of the flow, which is quite rare in biological systems, and not that easy to observe. And what we did is just to build a model where we start from a 2D cylinder representing the central canal. We implement cilia motile and with polarity towards the tail only on the ventral side. And the beating of the cilia is generating this force gradient that you can see here that tend to push the particles in the flow from rostral to caudal on the ventral side. And then passively, because the cylinder is closed on the two ends, you have recirculation on the dorsal side, so the particles are going in the opposite direction on the dorsal side. And with this very simple model, where the whole entire bidirectionality of the flow is explained by the cilia, you can recapitulate very nicely with simulation in yellow and green here, what we observed in data in gray in this image. So when you have sparse cilia, you actually get more area with recirculation. And if you have no cilia at all, you have no more flow, basically. And we could test with different methods to, close the the central canal on two ends and observed that the flow was still there locally, even though the central canal was closed, we could observe that this model seems to be really well accounting for the bidirectionality of the flow. So what could be the purpose of having a bidirectional flow at the embryonic stage during development? The idea that we promote is the fact that at this stage there is not yet blood flow, and that it might be a very effective way to transport big particles such as the large lipidic particles that we've imaged with the dynamic flow field OCT. Because when we do, again, simulation, we find that if you look now here at this graph with the cyan and purple, if we look at the time it takes to travel 300 micron as a function of the size of the particle, if you're a very small molecule, well, it's linear and it's not changing very much to have flow or no flow, because the regime is pretty much the same for small particles under 20 nanometer. But as soon as your lipidic particles are above 20 nanometer, which is typically the size of those putative exosomes that we've identified, then it makes a huge difference, because having the flow enables to travel much faster. And in fact, it's quite counterintuitive. The bigger you are, the faster you go, which is not what you usually see in biology. And the reason is that if you're big, you actually don't get trapped by the Brownian motion into the bidirectional, the two direction of the flow, and you tend to just stick to either the ventral side, where you go to the caudal end, or the dorsal side, where you go to toward the head. So we also noticed that muscle contraction were changing a lot the speed at which beads could propagate. So if we had paralyzed animals, the beads would propagate much less in the central canal than if you had active animal that could contract the muscle. And we observed that transport as well. So we've been looking at how does muscle contraction influence the flow. And what we notice is that every time animals, when they were free to contract, were contracting muscles during the typical embryonic twitch, where propagation in the muscle typically goes from rostral to caudal, we had a big increase in the flux of cerebrospinal fluid, and therefore in the speed of this particle that can be easily multiplied by 5 or 10 in our recordings compared to the typical speed we're observing. So the way we see it is that it's typically working like a peristatic pump, where if some of you have done recordings where you need to change the solution, you basically press in a direction against the tube in order to induce a flow. And we think that the muscle are in fact, doing this task and enabling the flow to be sped up by contracting along a wave like that. So every time there is a spontaneous contraction, the neurons, if we record them with GCamp, are showing very large calcium trunnion that you can see here on this image. So our original idea was that the cells were possibly just simply responding to the flow. But there is a problem with that. The problem is that you cannot really explain the observation of horse bone that the asymmetry of the activation. So when the animal bends the tail on one side, the cells that are recruited are only the cells from that side and not from the contralateral side. And talking to François Galère and Ludovic Kaiser, we understood that there is no way with just bending that you can change the flow in an asymmetric manner between left and right. The flow is null on the wall of the central canal, but it's not different on the left and right when you're passively or actively bending the tail. It's symmetrical. So we wonder what could be the reason why these cells are showing an asymmetry. And this is the moment where Adeline Horsdale-Imagine, who had done her PhD already with Nicolas van der Berg, joined the lab, and we investigated together the role of the Reissner Fiber, this large polymer that I've introduced to you, that we can see in this stack here. So it's the central canal in EM is very dense. We think that there are lots of proteins, and the Reissner, I have to play the movie again. The Reissner fiber is here, and you can see that it's very close to cilia from ependymal cells, as well as from the apical extension of those CSF contacting neurons, where we can see one here on the right of the image. So Aline did serial section and reconstruction of the CSF-CN and the Reissner fiber, and found that in fixed tissue, their apical extension was indeed very close to the Reissner fiber, and the cilia labeled here, you can see, is in close vicinity of the fiber. So it was natural to ask the question that Conner and Agger were disagreeing about, which is, well, could it be that this sensory cell form a sensory organ by forming a complex with the Reissner fiber? And so we tested this hypothesis, and that's the work of Pierre-Luc Bardet here and Yasmine Kanto-Belharif, who generated a mutant, in fact, two different mutants for SCOspondin. The first one that Pierre-Luc generated is a null allele, where we have used CRISPR in the first domain here in the ME domain and generated a stop codon, where we basically completely truncated the protein SCOspondin. And in the second mutant that we thought could be an hypomorph, Pierre-Luc observed that he had, instead of a stop codon, he had 5 amino acids added in red here that were in the ME domain, typically thought to be critical for the aggregation of the SCOspondin into the fiber. And so we then, in fact, it's Yasmine Kanto-Belharif who really investigated the properties of these mutants. And she discovered that in the wild type, as I've shown you, you have a very nice Reissner fiber that you can see from a lateral view here. And in the two mutants, the null allele and what we thought was an hypomorph, there is no more fiber. So there is absolutely no Reissner fiber material in the ICM13 mutant, the null allele. And in the ICM15, which has 5 amino acids added, what we observe is still accumulation of the SCOspondin Reissner fiber material in the floor plate, but not no formation of a fiber. And what is remarkable in these mutants is that the cilia are completely the same in terms of orientation, polarity, and beating properties, but also the flow is exactly the same. So with the same method I've shown you, Yasmine was able to record the movement of particles in the central canal here, and she found indeed exactly the same pattern of bidirectional flow for the control siblings, the ICM13 mutant and the ICM15 mutant. So the fiber doesn't seem to affect, at least at this resolution, the profile of the flow and the bidirectionality of the velocity profile I've shown you before. And this was confirmed by Olivier Touvenin in his quantification. So Aline went on to test, well, is the fiber necessary for the sensory function of the CSF contacting neurons? And she did different experiments, again, with active and passive bending, and find that in both cases, the response of the CSF-CN to the bending was completely abolished in the mutant compared to the control siblings. And here I'm showing you the ICM15 mutant where the fiber is not present, but we still detect SCOspondin in the cell that secreted in the floor plate. So we have evidence that the CSF contacting neurons require the Reissner fiber in order to sense asymmetrically the bending. And at this stage, we also have evidence from Laura Desbon, who was a grad student in the lab, that the apical extension, and in particular the length of the microvilli, is critical. So Laura did a series of different mutants where the apical extension was affected, and I'm showing you here the SPIN mutant, in which, as you can see on the image here, the apical extension and the microvilli are reduced, so their length is reduced compared to the control. And in these animals, the amplitude of the response is also reduced. So we have evidence that the sensory function of the cell requires the Reissner fiber and requires a full developed apical extension. And so this is like a beautiful movie from Ryan Gray's lab in University of Texas in Austin, where I'm showing you here. Ryan, and in particular Ben Trudvine in his lab, has been able to do a knock-in where he could make a GFP fiber to look at the fiber in vivo. And I'm showing you this movie, thanks to Ryan, to illustrate the fact that the fiber is under great tension in vivo. So Ben played with a laser and ablated the fiber in the center here. And what you can see is that the fiber immediately runs away on the rostral side, which is a sign that it must be attached in the area of the subcommissural organ and the flexural organ, and that as soon as it breaks, it goes like a rope. And that is really interesting. And together with our observation from the work of Adeline, what we think is happening is that the fiber is like a rope under great tension in the central canal, and when the animal bends on one side, especially during locomotion, it activates the cells by getting in close proximity to the apical extension. So we're trying to conceive experiments where we can image that now while the animal is moving, which is really challenging. But there are basically two hypotheses that we've put forward in the work of Adeline. One is that, and it's our favorite hypothesis actually, is the fact that you don't need to have contacts necessarily. You can have just a close vicinity between the apical extension and the fiber itself. And what is remarkable is that the flow of cerebrospinal fluid is null, which is shown here as dark on the fiber, because the fiber is very, very slowly moving compared to the particle that are in the CSF. And it's also null on the wall of the central canal. And so the gradient of flow that the apical extension is seeing is announced by the fact that the fiber forces a point of zero flow in the center of the central canal. And thanks to this null flow, you have a much larger gradient that the apical extension can perceive. So this is hypothesis number one. And hypothesis number two relies on the direct contacts between the apical extension, possibly the cilia, the kinocilium that we've observed, and the fiber. And we're trying to do experiments to test these two hypotheses. So now, what is the consequence of this on physiology? And starting, because we're a locomotion lab, what is the consequence on the locomotion of the animal? So we developed with Olivier Mira an algorithm to identify the kinematics of locomotion, where you can see we can identify each animal, their position, their direction of the head, and the position of the tail. And then we looked at different conditions. So we started with the PKD2L1 mutant, where we didn't observe anymore a recruitment of these cells during spinal bending. And when we quantified the tail angle over time during the escape, which is a typical massive response where you have very fast swimming and large amplitude of the tail bend, with a first C bend here where the animal is trying to go away from the danger, away from the aversive stimulus, that we're using an acoustic stimulus here. We observed that in the PKD2L mutants, the major phenotype was a reduction of the tail beat frequency, as you can see here in red for the mutant and in blue for the control siblings. And so reduced tail beat frequency means like reduced speed and less efficient at escaping from a dangerous stimulus. But there are lots of compensation in those mutants, so we investigated with other tools, and in particular, Jenna Stenberg, together with Kristen Severy in the lab, had been characterizing a very useful tool from the lab of Koichi Kawakami, where we could express the botulinum toxin B light chain in neurons, and found that it was very effective at blocking completely the release of neurotransmitter by acting on the snares. And so when we use this tool in CSF contacting neurons, we observed again the same phenotype of reduced tail beat frequency, reduced speed, and we also observed, something else, which I'm not going to show by a movie. We observed that the animals were now rolling more often when the secretion from CSF contacting neurons was blocked by the toxin. And so we were very puzzled by that because we really didn't know how to explain it and how to explain that sensory cell in the spinal cord could potentially affect active control of posture during locomotion. So compared to an animal that stays, I have it now. Compared to an animal that stays horizontal, in the control condition, we often saw the animal rotating in our recordings. And so two people have been key to start to understand how these cells were modulating motor function. Kevin Fiedler, a grad student, and Jeff Roberts done a recording where they were using Channelrhodopsin expressed in the CSF contacting neurons and recording from different targets in the spinal cord. And what they found together, so Kevin found that CSF contacting neurons were projecting onto premotor excitatory neuron involved in slow locomotion that are called V0V. And Jeff found that there were targets also in the escape circuit, in particular on large motor neurons involved with fast swimming and sensory interneurons that are critical for gating sensory information. So we had indication that these targets could be relevant to some of the phenotype that we've observed, but it didn't really quite explain the change of speed and the change of posture. The solution came from two more people who worked on this together in the lab, a grad student, Mingwei Wu, together with Martin Carbotano, who's a postdoc in the lab. And together they've tackled this question looking now at the CSF contacting neurons that are the most rostral in the spinal cord that project in the hindbrain, as it had been shown by Lydia Genoun previously in our lab. So what Mingwei did was to ablate the Rostralmost CSF contacting neurons here and do a control ablation where he was ablating the caudal one only, and another condition where he was not ablating any neurons, just mounting the fish the same, and then observing what is the consequence on the behavior. In this ablation, we've been very careful to make sure that we are not affecting other cells in the surroundings or other axons descending, such as the command neurons that are critical to tell the spinal cord what to do from the hindbrain. And so we verified this carefully. What they observed is the following. We observed that we had, again, a reduction of speed when we were ablating the rostral CSF contacting neurons and not the caudal one, as shown here. We also observed a slowdown of the time to peak, so an increase in the time to peak, and a reduction of the tail bend of the C bend. So less powerful and slower motion. And what we did to characterize this is to do deep learning to identify when is the animal rolling automatically in order to understand what was the kinematics of the defect in posture and figure out when it was happening in the behavior. And what we have understood is that when you remove those Rostralmost CSF contacting neurons, you don't affect rolling during the original C bend, the first bend here shown with the tail angle, but the probability of rolling becomes very large in the subsequent bend, and it's maximal in the third bend. So we have now an understanding that the Rostralmost CSF contacting neurons project on the brain, affect posture and speed. How do they do so? Well, they do so by projecting on two types, what we think of targets. First type is the cranial motor neurons in the hindbrain. So we have evidence from anatomy and electrophysiology that cranial motor neurons are receiving direct inhibitory input that are blocked by GABAzine, so GABA A receptor antagonist that are mediating this response. And we could label these cells by filling with a dye during the recording, and we found that numerous muscle in the ventral musculature are indeed receiving inhibition. So multiple motor neurons innervating this muscle are receiving inhibition. And we also found that descending command neurons, such as you see on the left here, the Mosner cell, which drives this large C bend, or on the right here, the V2 reticulospinal neurons, are receiving inhibition that we can characterize only with anatomy because we have evidence for axo-axonic synapse from the sensory cell onto the axon of the descending command neurons. In particular, we have apposition of the presynaptic terminals with the presynaptic button of the Mosner cell, suggesting presynaptic inhibition from the sensory neuron onto the descending command neurons. And we have similar evidence for the shock-stain, where we can label the postsynaptic site with gefferin and see that we have an overlap between the presynaptic and the postsynaptic target. So the schematic that we have in mind is that the CSF contact neurons can act via targets in the spinal cord, where they can affect locomotion at the, in particular, slow regime, and possibly also posture via this projection onto large primary motor neurons. But they have a critical role in the way they project onto targets in the hindbrain, with cranial motor neurons as well as reticulospinal neurons being receiving direct inputs from these cells. So now I think I've been quite long, so I will just give you the upshot so that I don't take too much of your time. We've really focused a lot here on the mechanosensory aspect, and we have a system that can detect bending of the tail and project onto key neurons from central pattern generator in the spinal cord, as well as motor neurons and descending command neurons in the hindbrain. So what could be the function of this system, and do we care? How does it really impact physiology? So we've been looking at two aspects in the lab. Yasmin Canto-Belarif has been investigating the developmental side. Does it matter for development of the body axis and of the spine? And Andy Prendergast has been investigating a new function, looking at chemosensory function, in particular in the context of infection of the CNS. And so really, I'll give you just the upshot. What Yasmin found was that without a fiber, animals don't develop straight, they develop curled. And in many animals that had defective cilia, we observe the same phenotype. It's been known in the field for over 20 years. In all these mutants with defective cilia, there is no more fiber. And work that she did, now confirmed by other labs, in particular Ryan Gray, Brian Tiruna, and Cheng Tan, is really indicating that this system is critical for forming a straight body axis. And Yasmin has been investigating the role of monoamine for modulating the shape of the animal, and possibly via interaction between the Reissner fiber and cells in the central canal. Now, there is another phenotype that we've observed. In the PKD2L mutant, we observed an increased curvature of the spine in the rostral spinal cord, as you can see here in the adult. This is typical hyperkyphosis. And there are evidence that this sensory system might be involved as well in idiopathic scoliosis. And so this, I'm just showing here the work from the lab of Cheng Tian, showing that in mutants where for receptor of the urotensin-related peptide that are secreted by the CSF contacting neurons, there are clear evidence for 3D torsion. And the lab of Ryan Gray and Brian Tiruna has now found more evidence that mutation in the Reissner fiber can also induce idiopathic scoliosis with typical 3D torsion of the spine. So the last point I'd like to make before concluding is that we have indication that the system is also chemosensory. So we've been doing a transcriptome analysis of the CSF contacting neurons and found numerous chemoreceptors. And we got the idea that these cells could be detecting the presence of pathogen in the CSF. So Andrew Prendergast tackled this question, which is really, really risky, and performed injection of a bacteria, Streptococcus pneumoniae, which is typically the main bacteria leading to meningitis in humans. And he observed a very strong activation of these neurons. And if we try to play with this cell, blocking their release with botulinum toxin or ablating the cells using the nitroreductase, we observe a very strong reduction of the survival of the animal. So we're investigating the path now to understand this mechanism. So to conclude, we have identified a polymodal interoceptive sensory system that is made of the Reissner fiber together with the cerebrospinal fluid contacting neurons. These cells are intrinsically mechanosensory in vitro, but they are way more sensitive if the fiber is there to announce the detection of curvature of the spine during active or passive movement. We believe that they are both mechanosensory and chemosensory, and that they can impact the kinematics of locomotion, posture as well as development and morphogenesis, with both body axis formation and spine morphogenesis being affected in the different mutants I've introduced to you. And we're now looking at the chemosensory function in the cells, in particular in the context of innate immunity. So I'd like to finish by thanking the people who've done the job, in particular the recent work of Adeline Ortsdal-Imagine, Yasmine Bellarif-Canto, Martin Carbotano, and Mingwei Wu, and all our collaborators. Thank you very much for your attention.
Thank you very much, Claire. It's the awkward moment where the whole room stayed silent, but
they were sleeping.
No, exactly. It's not because they were sleeping, it's because we're living in a strange world. But you would see, we have lost contact. Is it us? Wait, there's something weird. Indeed. You guys still out there? Hello?
Can you hear people when they speak? Or?
No, but I can see if there's comments. I lost the feed. That's very strange. But they saw you till the end, but something is strange here with the Worldwide Neuro. Are you guys still around? Can you write something down? Can hear now. We lost feed. Oh. Lost the last 5 minutes. Just back now. Didn't hear the end. You want to do the last 2 slides, Claire? Maybe it's worth it.
Okay. Well, we don't know when.
Okay, we're going to do that. Sorry about that, Claire. I don't know what happened. It's the first time.
Okay. I know it's okay. I'm not sure what the end means.
Well, acknowledgement for sure. You want to go. Let's say what people tell us. You had, I guess, maybe the summary slide.
Yes.
And then people that would want to go for more details can lost you in the middle of Andy's stuff.
Okay.
Meningitis.
Okay. Okay. Cool. So Andy's work is indeed like really, really original. So I'll just go through that slide one more time.
Sorry about the doublet.
No problem. So yeah, with Andy, we tackled this question of whether the sensory system being equipped with a channel that can detect change of pH might be involved in detecting pathogen, because we know that when bacteria are proliferating, there is an accumulation of lactate. So our idea was, okay, let's try to find out whether these cells could be recruited during an invasion of the cerebrospinal fluid by a bacteria. So we used a bacteria called Streptococcus pneumoniae, fused to M. cherry here, injected it in the hindbrain ventricle, let the bacteria proliferate. As you can see, it fills the central canal, and then we image the activity of the CSF contacting neurons. And to our surprise, after about typically 15 to 20 hours, we have massive activation that looks like nothing we've seen before of those of subset of CSF contacting neurons. And so we investigated, it's a long study, I'm really just going to the upshot, we investigated whether this matters, this recruitment matters to the survival of the animal by doing experiments where we ablate the CSF contacting neurons with nitroreductase, as shown on the right. And when we do that, we find that indeed, compared to the control here in orange, we have a huge reduction of survival of the animal when we ablate the CSF contacting neurons. So we are now investigating the molecular mechanism. How do they detect product of the bacteria, and what do they release to fight the bacterial invasion? And hopefully you will hear about this work soon. And then to conclude, I just said that we found a sensory system, thanks to the work of all these people who I'm very happy, some of them are in the room. We found a sensory system relying on the Reissner Fiber in interacting with the cerebrospinal fluid contacting neurons that can detect flow, but we have evidence also molecules in the cerebrospinal fluid. And in response to this mechanical and chemical activation, they can impact 3 things. The first one is that they can affect the amplitude, the power, and the speed of locomotion, but they can also impact the posture, because the tail, and it's true for many animals, is massive and very muscular. So a tail bend can strongly impact and rotate the head. And the system is really critical to influence posture. And in development, the sensory system is involved in the body axis formation, as well as in the morphogenesis of the spine at the juvenile and adult stage. And the last piece that I really just gave a teaser for, hopefully I can talk more about it soon, is that we have indication that these cells are also chemosensory, and via a completely different pathway, not involving PKD2L1, they can detect pathogen, in particular bacteria involved in meningitis in humans, and respond to it by releasing peptide in order to fight bacteria. And so I put my arrangement slide, thanking really from the bottom of my heart all the people who have been contributing to this really fun adventure that lasted almost for a decade now, and our collaborators coming from biophysics with Patrick Damas, fluid mechanics with François Geller, immunology and microbiology with Christina van den Broek, and the field of development and scoliosis with Brian Gray and Maya Lehtinen. Thank you very much for your attention.
Voilà, thanks a lot. Thanks for going through that again. Oh, wait, people didn't hear. You have to do it one more time.
And then I close the computer.
Yeah, so, I'm going to close your screen there, and you should see now, if things go according to plan, some claps, unless there's something really wrong. But it'll come. We have questions, and we can look on the side of the thing. Okay. There's a few questions. Please do type the questions within the voilà. Here we see them coming.
C'est la rentrée.
Yeah, exactly. C'est la rentrée.
Difficult for everyone.
We're getting back to their habits. So a first question here. This is by Alan Kay. This is a bit tangential, but does CSF in zebrafish flow out of the ventricles and over the brain as it does in mammals?
Yeah, so Natalie Urish actually has a beautiful paper actually showing the flow of cerebrospinal fluid. So I would refer to that. It's published in Current Biology 2 years ago. And she's really done a beautiful work to investigate this question.
Okay, and actually she's the one asking the next question. Is there a different decoding of chemical cues and mechanical forces postsynaptically to the CSF?
Yeah, so we think the mechanism that underlies the chemosensory function and the mechanosensory function for us seems to be very, very distinct. So the mechanosensory relies on the PKD2L1. It could also involve other channels, like Nicolas van der Berg showed, ASIC channel were involved in mouse. It's really possible as well in our model. But the chemosensory function, in particular for the detection of bacteria, is completely the recruitment of the cells are the same in the mutant for PKD2L1, and we have evidence that instead they are a chemoreceptor from the TACE2 family, orphan taste receptor that are involved for tasting the CSF.
Any speculation as to why the same cell would be both mechanoceptive and chemoceptive? Because it's really different functions, right? What would be the evolutionary pressure on that?
Yeah, so actually, when you look at it, cells are rarely just one thing, right? The sensory cells. So Laura Desbon and Julian Roussel in the lab have also found that lateral line hair cells using transcriptome approach and functional imaging are responding to chemosensory cues as well, even though we know for decades that they are mechanosensory. And if you look at it, the channel that constitutes the sensory function, those transient receptor potential channel, such as PKD2L1, which is also TRPP3, are polymodal. So they respond to many, many cues, and rarely, in fact, a single cue. So I think it's mainly us lacking curiosity in trying different stimuli on the sensory cells, because the DRG have been found as well to respond to chemosensory cues, even the one involved in proprioception, and are also related to innate immunity from the work of Isaac Hu in Harvard. So I think it's a general theme that cells are often sensing multiple things and need to be able to distinguish which one is which.
But how would the animal know whether the cell is responding to one or the other?
So in our hands, but it's really the beginning. The calcium response is very different. In one case, it's a channel and it's going through the membrane. In another, it's clearly a GPCR and it's recruiting like we think like calcium-induced calcium release. It's very different. And so I think it's really like, we need to go into the intracellular pathways to understand.
Interesting.
Yeah.
Okay. A couple of more questions. Do all CSF CNs, I don't know what that abbreviation means. Well,
so
CSF,
CSF contacting neurons.
Exactly. Okay. Right. At all rostrocaudal levels, project all the way to the hindbrain targets. Do you have evidence for recurrent connectivity between them?
So we were really excited by recurrent connectivity and didn't find any. We find that they project typically 2 to 6 segments above. And so when they are located in the first segment of spinal cord, which is segment 3 to 9, they will unavoidably project to the hindbrain, but they can also have contact with spinal targets on the way. So the neurons that we ablate are all going to the hindbrain, but they have also projection onto some spinal targets. So it's not completely, it's not a dichotomy, but clearly those cells are critical, and the projection that we demonstrate, the first demonstration of sensory feedback directly modulating descending command from the hindbrain, has never been shown before. It might be very relevant for mammals, because in mammals, the work from different anatomists identified that these cells project onto a region in the ventral fissure full of axons where there are no somas. So it could well be that they also provide presynaptic inhibition in the mouse.
I see. 3 more questions. Is the contribution of cilia to CSF CN activity altered developmentally or dependent on paralysis? So I mean dependent on movement. Seems different in result from CFAP mutant Sternberg 2018 versus Orts del Imagine 2020. Does that ring a bell?
Yeah, so there are like 2 lots of concern, and no. So in fact, what we shown in the work of Jenna Sternberg is that the CSF contacting neurons are mechanosensory via the channel, and in vitro they already have a mechanosensory property. But when I've indicated that we pushed with a probe that was 4 micron in a cell of 8 micron in the longest direction. So you basically punch the cell, you observe an increase in the open polarity of the PKD2L1 channel. Now, what we've seen in vivo is actually, even though they are sensitive to punches in vitro, when you don't have the fiber, their response to contraction and change of flow is minimal. So they really need the fiber to enhance the response in vivo. And that makes sense actually, because they are located in the center of the spinal cord, which is the location where you have the minimal deformation during tail bends. If you think about it, the side of the spinal cord are deformed dramatically. And in fact, Stan Greener did a beautiful work showing that there were mechanosensory cells responding, H cell responding to deformation there. But we find that they respond in the center, where from my calculation, the deformation is really under a micron. And so in order to detect that, they have to be coupled functionally with the Reissner fiber.
Okay. 2 last questions. Does the loss of CSF contacting neurons later in development, after 5 DPF, also affect the shape of the spinal cord or the shape of the fish?
So, sorry, what was the beginning of the question?
I guess if you lose these CSF contacting neurons later on, after 5 days post-fertilization.
So actually, it's an experiment we're investigating together with Ryan Gray right now. So we don't have the answer yet.
So what do you predict?
No, we know from the work of Ryan Gray that the fiber matters. Mm-hmm. And Brian Turina confirmed that as well in 2 publications recently. But we don't know indeed if the CSF contacting neurons contribute. So because we found a functional interaction, our guess is that it might be the case, and that's why we're doing these experiments. But we've been starting it before lockdown, and we're pursuing it. It's very tedious because you have to ablate the cell over and over again, they regenerate. And so we don't have the answer to this question yet.
Okay. Last question. You show that the CSF contacting neurons appear to have different functions. Would you say that different CSF contacting neuron populations are responsible at different spinal cord segments? I guess, yeah, it depends how you define the population. So maybe perhaps transcriptionally or...
Yeah, so what we see is that there are clearly two populations. The ventral one are actually activated by longitudinal contraction. That would be the crunches for us. And the dorsolateral one are activated by the left and right. And so these two populations are strongly distinct in terms of molecular markers. They express different receptors, different peptides, and in many senses, they have different targets. So our model that, hopefully will be published soon is that, in fact, they really enable different modulation on the slow and the fast swimming circuit. And the one that project on the fast swimming circuit are also the one that control the posture.
Okay. One last question that dropped in while you were answering. There are some publications saying that PKD2L1 opening is not sensitive to changes in membrane pressure, suggesting that other mechanosensitive channels are involved. Any idea about potential heteromers?
Yes, so as I mentioned, there is a beautiful work from Aline Orstelemagin together with Nicolas van der Berg, who investigated this question in the mouse in vitro, as well as Ten Grillner in the lamprey. And in both cases, using pharmacology, they found that the response of the CSF contacting neurons is strongly modulated by a blocker of ASIC channel. So one hypothesis is that there are indeed ASIC channels that could well be critical for the mechanosensory response as well. And we lose somehow the response when in the PKD2L mutant. So they have a way to interact either directly or indirectly with the PKD2L1 channel.
Okay. All right, we're reaching the end of the session. Claire, thank you again.
Thank you, Denis, for telling all those concerns in CSF-TNs and PKD2L1.
Yeah, I, as you can tell or imagine, I learned a lot.
Thank you so much for your
invitation. It's a back-to-school day for me too. Thanks everyone for being online. Next week we'll have Kevin Mitchell from Trinity College in Dublin. Join in and tell your friends to do so also. Have a good week until then. Bye-bye, everyone.